Yeah, thank you very much for the invitation to present some of our work on mucus and mucus as a barrier to drug delivery. Together with my colleagues and many students, we have worked on this topic for some years, and we are very happy that we start seeing some nice papers coming out. And I'll tell you more about these in a moment. But first of all, oops, I have to get the, yeah. First of all, there are numerous mucosal drug administration routes in the body, and of course that's the reason for our interest and for other people's interest in the field. And among these, we have the respiratory tract, we have the gastrointestinal tract, and of course also the vaginal and rectal administration routes. So if we take a closer look at, for example, the intestinal barrier that we are particularly interested in at the time being, we see, of course, that there are some morphological features of the gastrointestinal tract that are very interesting to study in detail, not the least the epithelial barrier with the tight junction proteins interconnecting the epithelial cells in order to promote and having a very tight epithelium in terms of not letting any undesired molecules or compounds enter the body. But what we also do have in the intestinal tract is, of course, the mucus that surface lines the epithelium, and that is there to lubricate and to protect the epithelium. Mucus is a viscoelastic layer, and very aqueous in content, and its thickness and composition varies throughout the digestive tract, just as it does throughout other mucosal surfaces in the body. So if we take a closer look at the mucus, as said, it's there to both protect and lubricate the tissue, and at a molecular level, the mucus consists primarily of mucin glycoproteins of different kinds that are interconnected at different hydrophobic interaction points, and that also consist of these cysteine-rich domains that are also interacting. So if we look at the content, most of it is water. The highest content of other molecules is mucin, as said, and that's the molecules that we are therefore most interested in. But we should not forget that mucus also contains a lot of debris and contents from, say, cells that have been excreted from the intestinal tissue and other tissues. So there are also lipids and proteins and other stuff in there. And of course, also the microbiome is present in the mucus. I'll not touch upon this today, but it's definitely there. So all in all, when we have this mucus barrier, we have first of all, we have a steric barrier that is due to the network composed by the glycoproteins that controls and to some extent inhibits the diffusion of large matter through the mucus barrier. But we also have an interactive barrier, so a barrier where, due to, for example, electrostatic interactions and hydrophobic interactions, that molecules can be maintained in the mucus rather than diffused through the mucus. And then, of course, also not to forget, we have a dynamic barrier, so continuously mucus is excreted from the body, and also that can constitute a barrier if one is designing a drug delivery system to cross or at least interact with the mucus barrier. So, as mentioned, the mucus is covering the epithelium. It's produced by goblet cells and excreted and lines the epithelial surface. And what is very clear and also summarized in this book chapter that we published not that long ago, it's very clear that there are a lot of different types of mucins, both surface bound and also secreted mucins. And also that these mucins are cleared within rather short time periods. So, of course, it's important to consider when we investigate a certain target in man for drug delivery to investigate the actual mucins that are present there. It might have an impact on the results that we see, and we'll get back to that in a moment. So for the work that we have done recently, we have characterized intestinal mucus properties, and overall, and also in accordance with literature, we see that the pH is around 6.5 in the intestinal tract. Osmolality values, of course, depend a little bit on the degree of hydration, but it ranges from, it's slightly hyperosmolar, at least what we have worked with, and ranges from 380 to 450 milliosmolar. There's a protein content of about 12%, and then a water content of more than 85%. So this is what we should keep in mind regarding the next work that I'm going to present. So if we are looking into drug delivery, which I think most of us in the audience are interested in here, of course we are interested in evaluating and affecting how drugs and drug delivery systems that may be optimized lead compounds at molecular form or dissolved at a molecular stage. And it may also be particulates or of nano size or larger size. But we are interested in investigating how these systems and also the functional excipients that are in the system can interact with the mucus barrier, maybe adhere, stick to the mucus barrier. So we are looking for mucoadhesion then, but also how mucus diffusion or mucus penetration may be achieved. So just before we get into the results, I'll just share with you a few slides on some of the thoughts that we have regarding the use of certain models. And what we have used for some years in my lab is that we have worked with intestinal mucus and from different animals, primarily pigs. And we have isolated that and characterized it in different ways. Here we have just an image of an intestine and also a cryo-EM image of the mucus that was isolated. We have also described a biosimilar mucus that is composed of commercially available mucins in combination with a few other components, the main one being a polyacrylic acid to induce the right viscosity of the biosimilar mucus. And we have done some work comparing the biosimilar mucus with the native mucus. I'll not talk a lot about this today, but just to mention. And then, of course, also we do work with cell culture models, both mucus secreting cell culture models, but also models where we combine these with our biosimilar mucus. And I'm fully aware that there are numerous cell models out there that are even more advanced than what is presented here. So these may also come into play in understanding how mucus works as a barrier for drug delivery. So of course, when we collect tissue, mucosal tissue, and also mucus, isolated mucus, we can use this for investigations by different methods. And some of the things that we have done is to look into if we use it in Using chamber setup, like setups where we have mounted the mucus as a barrier, and then we can apply a certain compound on one side and look into what is the appearance of the compound on the other side. Similarly, we can do this in transwell setups or setups that are similar to the transwells, either having the cells cultured in the filter insert or just applying the mucus at the filter insert. And of course, also diffusion chambers like the ones that are used for cutaneous drug delivery studies can be used in this sense. But that you can look further into when interested. So it's just to say what we have been doing. In addition to these previously mentioned methods, we have also applied different studies to characterize mucus, and some of these are very much based on rheological measurement, but also tracking of small particles within the mucus is something that we have done quite a lot to try and understand what's going on when the particle hits the mucus. I'll show some data on this in a moment. And then, of course, we have also looked at how does mucus appearance change, and how is it affected in the presence of excipients and nanoparticles, and that we have studied by QCMD and also by AFM. So before we go into the results, I'll just like to show this, what I think is a very nice figure summarizing strategies to overcome the mucus barrier. And of course, both size, as mentioned in the beginning, but also shape, surface properties in terms of polarity and charges can be determining for how well your molecule or particle of some kind, small or large, diffuse through the mucus. So that is something to consider, and there's a lot of literature out there describing some of these matters. Then, of course, also one may consider applying or co-applying mucolytics so that the diffusion of your compound of interest is increased throughout the mucus, and that can there are also different molecules that can be applied in this sense. Mucus adhesion may be pursued. I'll not touch upon this today, but it's definitely a way to achieve a higher or longer retention at the site of absorption, if that's an approach that you want to go for. But what is also not so often addressed, and what I think should be considered, is definitely to consider the pH, ion strength of the mucus, because that will definitely have an impact on how well this polymer network consisting of mucins is either expanded or collapsed, and that may surely have an impact on the diffusion through the mucus. So in addition to that, different ions may play an important role, and that is also something that we should and will be looking into in future. So for the next part of the talk, I will dig more into our results, and I'll show some data on the importance of size and charge in relation to viscosity of the mucus, and then also focus on particle diffusion in mucus samples, and finally end up with some data from research that was just published in JCR on the effect of certain excipients on mucus. So first of all, to start off with, the effect of molecular charge and size on diffusion. And as I already indicated, this is a steric barrier that can limit the diffusion of different compounds. And in this setup here, the way I report the results here, one of our postdocs did a study where she applied different molecular weight Fitch labeled dextrans from Fitch labeled dextran 4,000 to 150,000 to one side of mucus, and then investigated how fast this dextrans appeared on the other side of the mucus barrier that we employed. And we here, in this case, we used porcine intestinal mucus. That's the PIM abbreviation up here. And what is clearly seen, and maybe I should just hide this one. What is clearly seen is that with increasing molecular size, the permeability, apparent permeability value decreases, which is as expected. If we look at lag time, before we really see something appearing on the other side, we can also see that the lag time increases with increasing size. And derived from these two together, also the total permeated amount at the end of the experiment was relating very much to the PAP value, not surprisingly. Then what the student also did was that she looked into the effect of charge. So she used fixed dextrans with a neutral, positive, and a negative charge of the size of 4, 40, and 50, 150,000 Dalton. And it was clear to see that the positively charged compounds here, they had a lower diffusion through the mucous barrier than both the neutral and the negatively charged. This has also been shown previously with, for example, nanoparticles that of course are larger than even these molecules of relatively high molecular weight. But it was good to see that we could definitely reproduce and also explain these appearances in terms of apparent permeability. The lag time was kind of reflecting the same results, and of course also the final permeated amount was reflecting the PAP values. So definitely there is a barrier, a steric barrier, and also a barrier related to charge. Let's just go on. So if we then go further on with the diffusion studies, we had a small study where Ada here and Stine together, they looked into how to investigate the effect of viscosity on particle diffusion. And this is just, this first graph here is just to display the viscosity of a gastric mucin dispersion, so commercially available gastric mucin from Sigma at different concentrations. And of course, we see here that there's a difference between the very low concentrations of mucin dispersed and the higher concentrations. So this is just a reminder. So what we did was that Ada is working on catapulting spheres and controlling these catapults. So she worked with spheres of a relatively large size, polystyrene beads, and what she could see is that, let me see, I should be able to, hang on, apologies. So of course, the idea is that she, from the bottom, catapults a bead upwards. And if she does this in water with different powers, you can see that depending on the power, the speed is, of course, slower or faster. So that was as expected. But then when we used mucin instead of just water in the medium, it's of course clear that when you have some polymer molecules in the mucin dispersion, that the particle may not just take the direct path towards the upper level here. And then when we used this 1% mucin system and we tried to do the same, it was clear not only that the speed was slower, but also that the path was not straight. And if we put some numbers on this, we could see that the average speed was also, of course, dependent on the amount of mucin in the system. But a very nice visual demonstration of how this looks. So in line with that, we previously published some work where Feng had formulated some nanoparticles for delivery of antimicrobials. And of course, when applying these to the respiratory tract, the particles will also impact mucus and of course also biofilms in this case, but now we're talking about the mucus. So these PLGA nanoparticles, I'll just go back to my pointer. These PLGA nanoparticles were designed so that they were mucus inert, meaning that they had a surface coverage that would allow them to diffuse in the mucus, and then this covering molecules would be cleaved within the mucus, and by so the diffusion would decrease and be limited, getting some residence in the biofilm and in the mucus. So we prepared, very briefly, we prepared these PLGA nanoparticles, loaded them with azithromycin as a model antimicrobial, coated the PLGA nanoparticles with TPDS, and all this was done using a microfluidics system where we had, of course, several inlets, and also we characterized these particles by different means. So here the size was around 100 nanometers, and they were negatively charged. So together with the researchers at the science department, science faculty at the University of Copenhagen, we did some microscopy imaging. So we did turf imaging, and based on this, or when I say we, I mean Søren here and his colleagues, were looking at the tracking of the particles. So the idea is here that the focal plane is detecting the appearance of particles that diffuse from a surface of a mucin dispersion, and then reaches the focal plane and the objective where they are detected. And it would look something like this when looking from the bottom of a glass slide where mucus is mounted on top. And based on this real-time tracking, it's possible to determine the spatiotemporal localization of particles and number of particles within a certain frame. And this we would like to use to investigate to which extent certain particles actually do not interact with the mucus surface, or they diffuse directly through, or maybe they take a certain path. So with the particles that Fung developed, we did a lot of tracking of particles and looked at the trajectories as well. And overall, what we found that when we had all these trackings and recordings, we could determine that the step lengths here, on the x-axis here, definitely increased when we had the TPDS coated nanoparticles, so the nanoparticles that we designed to be mucus diffusive. And we could see that the step lengths increased significantly, and we could also, of course, see that the diffusion also increased, meaning that more particles per frame of the TPDS coated particles were higher. So when we take this on to display it as a hotspot appearance, or when we looked into hotspot appearance and distribution of particles over a course of time, we could see that the non-coated particles over here, they kind of, at least for the first 1200 seconds, did not really diffuse very well. So they did not appear at the surface of the glass slide. Whereas after a certain time period here, 1200 seconds, it started to increase in rate, and also these particles tended to appear at the surface. However, if we looked at the surface coated, as already indicated, we could see that there was already from the start of the experiment a rather fast diffusion through the particles, through the mucus, and that also was maintained throughout the experiment. What was also interesting in this case was to see that if we started looking for hotspots, so the areas where there was a high level of red staining, we could see that these hotspots were conserved to some extent, at least, in the systems between the first recordings and the later recordings, meaning that maybe there was some kind of tendency towards a heterogeneous distribution of diffusion of particles from the surface to the detection level of the system. And of course, this can indicate that particles do not only follow one straight path through the mucins, but there are maybe areas that are more prone to leading diffusion of particles through the mucus rather than others. So in addition to these studies, we often apply QCMD in order to, so it's quartz crystal microbalance with dissipation, in order to investigate to which extent do material interact with mucin that is, for example, associated to the surface of a sensor on the QCMD, and also how does it interact with the material on the surface. So we can, by this means, we can detect the thickness or the amount that is associated to the sensor, and also to some extent investigate how soft or how rigid this material then becomes. And very briefly, I'll just show that Feng, he did some work where he applied or conjugated mucin directly to the gold sensor. And this is all described in a paper a few years back. So he conjugated mucin to the surface, and that is commercially available mucin. And then he applied the particle. This is the non-coated particle. And he could see that after applying the particle, that the frequency dropped, and also that the dissipation leveled out. Then the interesting part is, of course, to see what happens if you then apply the coated particle. So this graph over here is similar to the one I just showed you, except that the colors are opposite. I apologize for that. But that the frequency drops and the dissipation levels out. When we have here the coated particle, we see that the frequency decreases very, very slowly, meaning that the particles do not adhere to the surface and do not contribute to the mass on the sensor. And then also we see that the dissipation increases slowly. So when we have data like this, we can relate them and look into how is the relation between the dissipation and the frequencies, and that will give some kind of indication as to how rigid or how soft the layers are. And I will not spend too much time on this at this point in time, but what I'll say is, of course, and point out that you will see that there are differences between the bare particles and the coated particles. So definitely these kinds of methods can also be applicable to give some insight into what is going on when particles, in this case, are interacting with mucus. So the last part of my talk will be on the effect of excipients on mucus, and this is primarily the work of a PhD student, now postdoc, Janni, in our group, who did a lot of work on mucus, and what she did was that she primarily used porcine intestinal mucus and also did some work with gastric mucus. And I'll just remind you here that, of course, these types of mucus is constituted by different mucin molecules that may have different properties. So Janni was interested in looking into how is the effect of lipid-based permeation enhancers influencing the mucus and the diffusion within mucus. So she, of course, considered different types of enhancers, and in the end, she chose to work with the SNAC, so the salcaprosate, that is the excipient in the Rybelsus product from Novo Nordisk containing semaglutide as a active drug. And she used rheology to a large extent to investigate how SNAC in some titrated amounts, but here I'll report only the 150 millimolar, how that interfered with the rheological properties of the mucus from both intestine and also from the stomach of pigs. And to start off, she showed that the SNAC, the contribution of SNAC was that it increased the viscosity of porcine intestinal mucus, whereas it seemed to decrease the viscosity of gastric mucus. And of course, we are very well aware that there's a high variation here in these data. However, that's of course the condition when working with biological material, so we cannot exclusively conclude that there is a difference here, but we can definitely conclude that there's a difference here in increasing the viscosity of the mucus, which was kind of contradicting what we had first thought. So also, if she then further investigated the effect on the storage modulus and loss modulus of the mucus, she could see that there was some kind of the same tendency between using intestinal mucus and gastric mucus when applying 150 millimolar SNAC to the mucus samples. It was clear that the amount of intermolecular interactions increased by the light gray bar and the light blue bar here. And also that seemingly the strength of the intermolecular interactions increased. Nevertheless, the overall effect on the viscosity was different between the porcine intestinal mucus and the porcine gastric mucus. So what Janni continued to do was to do a diffusion study using some relevant peptide drugs, cyclosporine and valbamine is not peptide, but it's a hydrophilic macromolecule. And she did this in the transwell setup where she applied mucus in a setting here and then applied some drug of interest on the one side and took out samples on the other side and analyzed for the amount permeated. And if we just look at the amount permeated, it was clear that in both these cases that actually when the SNAC was applied, there was a lower amount permeating across the intestinal tissue, intestinal mucus, as compared to a less significant, if any at all, difference in diffusion in the gastric mucus. So this we found quite interesting, and I'm happy to say that this is published in Journal Control Release and is online now. So you can go have a look into further of the data. So of course, the point is to understand what's going on with the SNAC interaction with the mucins, and this may of course be related to changing the electrostatic repulsions between the polymer chains of the mucus, so the mucins themselves, and this is definitely something that can explain why we see this condensing of the mucus in the presence of SNAC. If we look into the amount of especially sialic acid that is negatively charged terminal groups or units on the mucins, then we can see that in the intestines the amount of sialic acid is higher than in the stomach mucus. This is, of course, I should note, reported for mice. But this difference may explain that SNAC seems to have a more significant effect in the intestinal mucus than in the gastric mucus. So Janni also did some cryo-EM imaging of the mucus before and after exposure to SNAC. And this is the intestinal mucus that has been exposed to 150 millimolar SNAC. And what we can see when comparing to the control, we can definitely see that the pore sizes seems to decrease. Also, of course, as a result of the collapsing of the mucus. And that is, of course, well in line with the fact that we see a lower diffusion across the mucus that has been exposed to SNAC. So to top off, Janni also did some particle tracking, and here it was nanoparticles of around 250 nanometers, where she looked into how did these nanoparticles distribute and in a mucus sample. And here we have heat maps showing diffusion in control mucus and diffusion in mucus that was exposed to SNAC for a certain time or period, and it's very clear and obvious that the distribution is narrowed when in the presence with SNAC. So the diffusion in the mucus is decreasing in the presence of SNAC. This is also reflected from the alpha values that is reported here in D and E, where you can see that the diffusion in the absence of SNAC, so in the control mucus, is higher than in the presence of SNAC. And that's, you can also to some extent see from the diffusion, cumulative diffusion coefficients and viscosities. So all in all, this comes back to the previous study that I talked about, that particle tracking can indeed help explain some of the effects that is occurring in mucus when exposed to, for example, excipients. So to conclude on the last part, I want to conclude that the lipid-based enhancer SNAC, in this case, indeed changed the properties of mucus. It changed the viscosity, G prime and G double prime relationship, and that correlated to a decreased permeation of two compounds that we investigated, cyclosporine and valbamine, through the porcine intestinal mucus. And also, interestingly, we found that the effect of SNAC seemed indeed to depend on the regional site that the mucus was isolated from, whether that was the stomach or from the intestine. And of course, this primes us to do more in order to understand more specifically how enhancers and formulations influence the mucus barrier, and how this can be tailored in terms of achieving a better drug delivery. So before I end, I'd just like to summarize the talk by saying that we indeed think that mucus constitutes a specialized barrier to drug diffusion and also nanoparticle diffusion, that excipients may affect the properties of mucus, may very likely affect the properties of mucus, I should say, but also what we trust is that we have to really be picky on the models and the orthogonal methods that we apply in order to be able to conclude specifically on the mechanisms of actions which we are interested in order to solve and improve drug delivery via the oral route. So with that having said, I'll just thank some of the key persons and places involved in this work. Janni, as said, was one of the key people generating data for this presentation, and also Fung, and then also Ada at the DTU in Denmark, and together with, of course, collaborators at the nanoscience department. And of course, none of this would have been possible without funding. So I'd like to thank especially the Novo Nordisk Foundation for this Grand Challenge grant that we are supported by for the moment, but also previous funding from IMI in terms of this compact consortium, and also, of course, regional supports from the Research Council. So with that, I'd like to thank you all for your attention.